barges and other means of transfer. These should be avoided. Colonization, is it required? I don't think so. We already mentioned that the microorganism can be a temporary colonizer, it can even be a transient, non-colonizing microorganisms. And when we go back to the drug resistance again, do we really want the microorganism to be sensitive to absolutely all antibiotics? Just 5-10 years ago, the answer would be definitely yes, we want them to be sensitive. Now, I would say it depends. It depends on the specific scenario where this probiotic will be used. What if it is going to be used in combination with antibiotic therapy? What if it will be used just after the antibiotic treatment is finished and some residual amounts of antibiotic is still present in the eukaryotic organism and you don't want the probiotic to die? So it really depends. Bile salt and pH stability, certainly yes, if you're talking about the gastrointestinal application. And even the pH stability may not necessarily be limiting thing. Why? Because nowadays we have very powerful delivery vehicles which serve to protect live bacteria from the harsh environment through which the microorganism has to pass on its way to the site of action. And when we look at each of these steps, fail, reject, fail, reject, fail, reject, which means that if we will follow this scheme by the book, each and every step, we may check many microorganisms that have great potential, but do not fit one or another parameter. So these should really be approved. When we look at the schematic representation which summarizes the omics platforms and how they can provide the information that is found useful in our hard job with the selection of potentially probiotic bacteria. One thing should be mentioned: we will have a standalone dedicated session where we will be talking about omics, and this is where we will dive deeper into these issues. But I want to go through some of the issues that we have on these slides in no particular order. We do need to know how the health benefiting microorganisms are related to each other. We needed to find similarities. We needed to identify the areas where their application can be most useful. We need to know and to understand if they will be able to resist certain conditions that may face in the environment where they have to work. And of course, not all of them, just some mentioned here. And of course, gram-positive, gram-negative, obviously we know how different these kind of microorganisms. We're going deeper into the issues related to each of them. Faculty anaerobes, strict anaerobes, which is again very important thing. How do you deliver these bacteria to the site of action? How do you protect them from the environment before they will come to the site of action? And many other things. If we will look at the omics and how they can help, and again in no particular order, just randomly, looking at some things here in this pie chart, bile salt stress and tolerance to bile salt and the human gut adaptation. Of course, they are important for the probiotics that are aimed at the gastrointestinal tract, and we do know that majority of probiotics that are currently being investigated and marketed both as over-the-counter products and for medicinal application are aimed for the gastrointestinal tract. And that's where we are dealing, for instance, with the information that can be delivered through transcriptomics, proteomics, and many other areas. Cold stress tolerance and action. You may ask, why is that important? Well, what if you are dealing with the microorganisms that are expected, for instance, to work in the aquacultures of the cold water condition environment? And we do know some examples of probiotics performing beautifully on aquacultures in warm water reservoirs, but not being really that active in the fish that was grown in the cold water environments. So these are important factors. Another area where we can get some knowledge with assistance of metabolomics, for instance, production of bacteriocins. I can't resist mentioning it because we will have a special standalone session dedicated to bacteriocins as one of the powerful weapons at hand of the health promoting bacteria. And so knowledge of the ability of the microorganism to produce bacteriocins empowers us with understanding of how these microorganisms may combat the intruders, the pathogenic or spoilage organisms in the environment, whether we are dealing with infection diseases or with the microorganisms that may spoil food products. Now, this is where we have an example of the multi-omics approach, and well, we have big here, but of course these can be extrapolated for many other hosts, eukaryotic hosts, and I want to apologize and at the same time explain the audience one thing. You will be having a lot of text on some of the slides, and the reason for that is to make it easier for you, so you will get the quick essence, the most interesting and important information related to the visual representation extracted from the publication, so you will have it at hand by the side of the picture, right? So this approach was actually adapted originally from the multi-omics filtering protocol that was developed by Remoller and his colleagues in 2016, I believe. And that was done for the amphibian skin. And then it was, they realized, the researchers realized that this approach can be used for many animals, such as monogastric pigs and other eukaryotic hosts. And the meta-omic databases indeed are a very valuable source of information during data mining. You can see along with the step P elucidation that happens at the step C, and monitoring of fever conditions trials as outlined in the step T. And these help to identify the marker genes, proteins, and metabolites in the microorganisms that are being selected as potential probiotic strains, making it more convenient to set up the genetic and molecular selection processes for potential probiotic trials in the future with the laboratory animals, for instance, and then scaling up with animals of agricultural importance. So what kind of information we gather, for instance, from sequencing of the genomes of the gut bacteria, that provides us with a library of the genes discoveries. Kleinheimer he passed away some years ago. He was one of the gods of bacteriosis genetics. He was one of the first who started diving into the research on probiotic bacteria, especially being focused on lactic acid bacteria. And his group was one of the first to derive a so-called minimal gut genome from the catalog of the genes of human gut microorganisms. And that at that time, I think it's somewhere in 2010, 2011 perhaps. And in these genes, there were several essential ones identified, such as, for instance, the ones that are involved in the carbohydrate metabolism, the ones that are essential for the adhesion function, the ones that are involved in the interaction with the gastrointestinal epithelium, and the specific housekeeping genes are essential. They're required as the core genes that function and which can deliver the health promoting function, but at the same time allow the microorganism to survive in this environment. Metaprobiotic analysis combines the quantitative real-time PCR and DNA fingerprinting and shows that, for instance, lactobacilli can actually influence the changes in the weight, but also this analysis allows us to see the difference between different species and even different strains of lactobacilli involved in the processes. And culture-independent metaomics approaches can also help us with linking the proteins and the metabolic pathways to the specific functions and the properties of the microorganisms that are selected as probiotic candidates. And these silica methods, they can also help us to provide with the functional assessment of the candidate probiotic microorganisms. And then we can obviously go from in vitro to in vivo and ex vivo analyses. We can go from the small scale to the larger scale controlled experimental trials. Why is that important? All of what just was mentioned, two reasons. Number one, understanding the mechanism of action, and through the understanding of the mechanism of action, we can develop the most effective way for these microorganisms to deliver their function. And of course, it will save a lot of money if we will do all these studies and if we will prove that the microorganism indeed has some potential before we move to the large-scale clinical trials or large-scale animal trials, which can be pricey. Probiotic selection criteria. On the left, I think it is somewhat over-engineered, but I cannot blame the authors because you really want to put everything into the picture. On the right is the bubble that I put together, which I think is kind of an extract from what we see on the left. I already mentioned before that the systematic approach is the must. And the systematic approach is required, and it's done using the strategy that is similar to the one that is shown, on this figure. And in most cases, we have many strains isolated, which are considered to be potentially health promoting, and then we have to narrow, and the selection is being narrowed through the step-by-step approach consisting of a sequence of tests to reduce the number of the potentially health promoting microorganisms by excluding those that cannot deliver certain functions. And at the end, we will have the selection of fewer strains that mentioned it before, and I think this is crucial when we are designing probiotic or selecting probiotic for action in a specific environment, in a specific eukaryotic host. In vitro performance, in the lab medium, in the targeted animal simulating, and I cannot tell you how many times we see examples of the microorganisms being tested in the ideal lab environment without any tests being conducted in the environment that is simulating the animal or human body conditions. Why 37 degrees C? Why rich medium such as, I don't know, amaranth and some other conditions? Then animal studies, of course, and studies of the production and storage performance. We're now moving to the safety. Safety was already mentioned several times, and the known and unknown about the safety, they do matter. And I will give you some examples from humorous to tragic. And I think that is important. We have to be conscious. We have to be careful when we are selecting microorganisms. We need to know who they are and where they're going. And of course, just to put a smile on the face of audience, I know the time is getting to the evening at your location. My day is just starting, so both of us, myself and the group, we need to put a smile on our face. And God knows why some scientists decided to feed these cute little creatures with yogurt and to measure the size and the weight of their testicles. But they found, what a surprise indeed, that the size and the weight of testicles of the mice fed with yogurt were bigger compared to the control group. Okay, even from these humorous examples, some take-home messages should derive. And I guess the take-home message here will be that health-promoting bacteria may act in so many ways, and sometimes not even the ways that we expect them to act. And we have to be prepared to analyze every possible outcome of the use. I'm giving you here the picture from the old cardboard of Activia. Actually, I still have it with me in my office, and I always show it to the students. It's not something that I created. It was Activia. It says here, it's scientifically proven to help reduce long intestinal transit time. And, really, do we want to reduce this time if we're otherwise healthy? And sure enough, there were cases reported for Activia causing diarrhea in the past. And there was the legal case built against Activia. And, everything disappeared in the thin air. I'm one of those who suffered because as a real scientist, I wanted to try this product. I never took it before, but I did try, and I suffered from the outcome. And as a real scientist, I repeated the experiment 3 times with the same outcome. So I still want to face the Activia guys, the Danone guys, and to ask them the question, what's wrong with that? We also know examples of the stool transport going bad. Of course, in the majority of the cases, the health benefit is delivered and well documented. But there were a few, not just single, but a few cases reported when, by mistake, the sample from the obese individual was delivered to another human subject, and this human subject developed tremendous weight. And unfortunately, these did happen. So one should be really very careful, and that is the message to those who are the companies who are providing service. Please don't get me wrong, it's really a very powerful method to deliver health promoting action to otherwise, non-appeased but still requiring some assistance individuals. The samples should be carefully analyzed and properly stored. And saying that, I want to turn the page and now to tell you about some probiotics that actually act as the mold in the sheep's skin. And one of the very sad examples is what I call the clinical trial that was conducted in early 2000s. And basically what happened was that the individuals with the severe acute pancreatitis were delivered probiotic bacteria, and at that time it was reported in the publication in Lancet that probiotics did not reduce the number of infections but resulted in a high number of deaths. Later, the statement was debated, questioned, and there were, I think, a couple more papers published saying that if to conduct the statistical analysis using different tools, you can see that there were no bigger number of deaths in the experimental group. I'm not going through these kind of things. The most important things here are, well, first of all, should we really be conducting human trials on individuals who have their immune system imbalanced and declined in its activity? I don't think so. Another important take-home message from here: there was no reduction in the number of infections. So the microorganisms that are performing as good fighters against infectious bacteria in the test tube, in the lab environment, perhaps even in the animal trials, did not deliver their action here. Why? Could it be because the eukaryotic host was unable to help the health promoting bacteria? Could it be that the imbalanced immune system was acting not only against the host but also against all the microorganisms that are residing in these hosts? We don't know. But of course, this case raises concern and raises questions, and these questions should not remain unanswered. And whenever we design new probiotic, new application, we should be asking ourselves these questions. And with that, I'm turning here to another table with examples, and we have here what is commonly known as LGG. You will see this acronym, this abbreviation in the marketed products, even in the publications, Lactobacillus rhamnosus LGG strain, and it may cause sepsis. All right, and when you look at this table, one thing for sure catching my attention: the age of the people. We can see either very mature, not old, 74, 67, which means that the immune system is declining, all right? And we see those little creatures, which is less than 1 year old. I don't think that the immune system is fully established in these individuals. It's close to, but not quite. So this raises a concern. Could it be that the eukaryotic host was not simply fully protected against intruder, and whether you want it or not, but what happens with the delivery of probiotic bacteria, it's an act of intrusion, and Some may say that I'm a bitter, cynical, and skeptical person, but quite frankly, I don't believe in microorganisms being fully healthy and fully friendly. My personal take on it that microorganisms can be conditionally healthy. And as I said at the beginning of our today's gathering, they're making deal with the eukaryotic host. They're asking the permission to stay, and they're promising not to behave badly. But when the host is weakened, many things may happen. Another example would be with the spore-forming probiotic Bacillus subtilis, which can cause bacteremia. And we see here only one risk factor identified with the neoplastic diseases, not stated for the individual 79 years old. Chronic lymphocytic leukemia is stated for 73-year-old individual. I'm curious what went wrong with the 47, 25-year-old people. Most certainly there was something wrong with their health. So take-home message: if you want to deliver probiotic, and if it is delivered as a medicinal formulation under the supervision of a medical practitioner, of a medical professional, the health status of the human subject must be thoroughly investigated, and it should be known. Otherwise, if it is an over-the-counter product, every person is on his own, of course. Choosing the right probiotic and clinical decision algorithm is very so. One thing, and you will see these kind of, pretending to be handwritten comments appearing on slides once in a while. And I'm trying to put notes for myself. Treatment versus prevention, huge difference. And the important thing, and again, so many times you read the research paper and you see the design of the experiment, and the researchers, the investigators are talking about drug development or application to treat infections or something else, and when you see the scenario of the experiment, you realize that this is not treatment, this is prophylactic measure. So this is very important. It must be properly developed. All right, and of course, the things that have to be observed. The specificity, the identification of the area of application, the strains of the microorganism should be specified and identified. And of course, the quality of the product is the most. And last but not least at all, the access and regulatory bodies that are taking care of safety of the end user. And of course, when we're dealing with the medicinal formulations, it should be the FDA disclaimer, and the probiotic microorganism should be listed as a species and as a strain. And, go to the supermarket, look at the over-the-counter dietary supplements, and especially the food products that claim to be probiotic. How many of them have the probiotic bacteria identified to the level of the strain? Not all of them, unfortunately. Right? So these are the important things, because ultimately they allow us the traceability of the strains of the formulations whenever it is needed. So, is it a probiotic? Is it a drug? What are the functions that are delivered by these microorganisms? And we see that some of the functions are broadly presented in a variety of probiotics, such as the weak organic acid production and the short-chain fatty acid production, normalization of the resident microbiota. Some are frequent but not common. Production of vitamins, noxious microorganisms or activity of certain enzymes, such as, for instance, I don't know, we see bacillus natto, the production of nattokinase, and some of the functions that may be considered as rare functions, specific endocrinological, neurological effects, specific effect on neurological, on the central nervous system, production of specific bioactive components and define specific targets. But each and every time when we're talking about these things, when we're moving on the bumpy road from the fermented food containing health promoting bacteria to the drug, the side effects, are there any, and how many of them? They were proposed in vitro safety assessments, and you can see some are indicated with an arrow. I did it, just to make sure that we know what we're talking about here, and for myself to remember, hemolytic analysis is done virtually by every group that is working on isolation and identification, characterization of the potential probiotic bacteria. Presence of antibiotic-resistant genes, again, something that is done pretty much by everyone. Other issues may or may not be done during the in vitro analyses. Agglutination, co-agglutination, adhesion, bile salt deconjugation, presence of microcapsules, etc., etc. Safety assessment, what to say more? And of course, these are important things, and I put bubbles to highlight the most important or most interesting things. Of course, there should be no risk with using these probiotics in regard to antibiotic resistance, in regard to the possible action of virulence genes and traits, and we discussed it just a few minutes ago. And of course, it is important to test on the possible toxin production, hemolytic activity. The absence of hemolytic activity, of course, is a must. Side effects, the absence of the side effects is important indeed. And of course, the establishment of the history of safe use in traditional products. That is in the case when the probiotic microorganism is isolated from the traditional product. Formulations and the safety assessment, and again, a list of several bullets, and I'm not going to walk through all of them. Some additional components are mentioned here, such as possible allergenicity, the understanding of the dose essential for the activity, and the microorganism should not be overdosed. And we do know several examples, especially in the animal trials, when overdose with probiotic, for instance, can cause diarrhea. Right? There should be the record of the isolation history and taxonomic classification. The manufacturing process should be thoroughly controlled. And all these conditions are the must to deliver the safe product for the safe use by the human subjects, and of course in the animals of agricultural use. We're coming now to the conclusion part of our study, and I just want to give you some examples of probiotic strains and their use in prevention and treatment of some diseases. Diarrhea can be controlled by numerous microorganisms, and we can see here LGG strain, the very well-studied Bifida PP12. Some of the Saccharomyces thermophilus can also act there. Inflammatory diseases and bowel syndromes, again, we can see LGG is performing really well. Modulation of the immune system. I want to point to Lactobacillus shirota. We will be talking more about these microorganisms in time. Some of you, if not many of you, may know Yakult, which is the fermented dairy product containing Lactobacillus shallota strain. Prevention of allergy, and again we see here the well-studied and marketed PP12 strain of Bifidobacteria and Lactobacillus GG strain. So the take-home message from here is that yes, indeed, these microorganisms can perform well, and not only on the basis of individual uptake as the dietary supplement of the counter product, but also in the clinical trials we see some positive outcomes. These microorganisms are marketed by many companies. Some of the names you should certainly know, like Nestle, Wisconsin, Daniska. Well, now it's Daniska is a part of the DuPont company. Yakult, I already mentioned, by GIAN, Sweden, is also a very well-known manufacturer of probiotic material for a variety of companies. Just to finish, whenever I do brainstorming sessions, and unfortunately we will not have it during these gatherings since I'm far away from you, but I hope one day I will visit and maybe we'll meet with some of you. So what is important really when we're talking about beneficial microbes, we need to understand the role of microbiota and microbiome in general. We will be talking about microbiota and microbiome in one of our sessions. I already mentioned that I think that they are conditionally safe, so we must be very careful when we're designing formulations. We must know that they will not turn ugly when the immune system or the health of the human subjects or animals is compromised otherwise. We need to understand the interactions between the host and the probiotic microorganism. And of course, in capital letters, number one, safety, 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 safety must come first. Turning to the last slide of our presentation. So what we need to know here is what is the way, what is the path that we're taking. We're first screening and selecting, we're then going through the understanding of the role of potential microorganisms that are considered as probiotics in the natural environment, in the environment where they're going to serve their job. And finally, we need to understand how to produce them on the large scale for medical, agricultural, and veterinary applications. And finally, just to finish our today's first gathering, of course, with some humor, if the science of probiotics hold clinical trials, yogurt would replace many pharmaceutical drugs. And I always ask, students, especially the younger generation, when in the summer you're getting the sunburn, your grandmother, is she once in a while suggesting you, instead of using some medicinal, cream or whatever, just to put on your burned shoulders or face a little bit of yogurt? Because that was the habit, my time when I was a child. And with that, I would like to thank you all for our first gathering. We'll see each other again, I believe, in 30 minutes. Dr. Twari.
It's fine. Prakshita and Das, thank you very much. And this is up to you. If you want to take a break of as far as you are comfortable, you can join back.
Right. I think it's up to the audience also. But I will be back. I can be back like in 15 minutes. That's fine with me.
Yes. So any one of you want to interact with Professor Chikintas for a while in a quick question? So otherwise,
Sure.
Otherwise, he will go for rest and we'll join in 15 minutes.
Any questions are welcome. Okay, please, whoever raising hands,
You can unmute and ask the question if you want.
yes.
So they are more interested in listening, I think.
Well, it's a beginning. We're just starting.
Beginning. Yes.
Right.
So, it is an instruction for all the participants that as per the instruction of GIAN national body, you have to join this course by name and your attendance is compulsory to get a certificate, and there is an examination at the end of the scheme, at the end of the lecture. So you have to pass this examination to get the certificate. So be careful whatever lectures are going on, and be interactive, and be careful to gain something out of it. Not because of fear of examination, but because something of eager to have a knowledge out of it. So
And I believe the participants will get the handouts. Is that correct? Dr. Tiwari.
Many of us cannot unmute. Or you cannot unmute yourself.
Well, there were issues with me in the beginning, but I can answer some questions that I see written here. If all the marketed probiotics are lactobacilli, no, certainly not. And I already mentioned today, some of them belong to bifidobacterium, some of them are lactobacilli, but of course we have to keep in mind that majority of marketed probiotics belong to lactobacilli and bifidobacterium. And that is something that historically happened because these were the first microorganisms. Of course, there are many probiotics in the group of yeast, Saccharomyces cerevisiae. Of course, we should say, and we will be repeating it again in the course of our sessions, the first and the most broadly known and the first technologically produced, I believe, if I'm not mistaken, was Escherichia coli. The E. coli Nisl in 1917 was the first probiotic, and obviously it's not lactic acid bacteria. Nowadays, the emerging group of probiotics belong to spore-forming microorganisms. And so lactobacilli were perhaps historically one of the first, and they are still dominating the market.
Yes, Dr. Chikindas, actually there was some technical issue. They were not able to unmute themselves. So now it is open.
Right.
You want to participate in this question answer, can ask.
Sure.
Unmute and ask. That should be very quick.
But they can also, the audience, respected audience, you can also write, they can see what you're writing here. If you have any questions to ask verbally, just ask verbally. Well, I guess that was the only question we've had so far about the production, the types of the microorganisms, the species involved. But we're just starting and more things to go through. So we'll have a brief break, let's say 10 minutes. And for you, it will be, in 10 minutes, right, Dr. Tiwari? What time will that be?
No issue, sir. Whenever you are comfortable, it should be, because I don't want to make you more tired. Take a rest and then start other one.
All right. No, I'm not tired actually. If you don't mind, I will just have a gulp of tea, just a little bit, and I can probably continue. I still have to teach today in the afternoon. So I'm not on the podium.
Yes.
Right. All right. I'm stopping the slide sharing, and the second one. And this is where we'll be talking about the probiotics for gastrointestinal health. And
Yes.
let me see. Again, as I said, my apologies. I'm still learning how to use it. And share. I can see that it appeared.
Yes, it is.
And I wonder if you can see the pointer, the arrow.
Yes, it is visible.
Okay, fantastic. So
It is visible.
if the group is with me, let's start, working with the group. And my apologies, I still have a little bit of tea. Goodness, nothing is better than a good tea in the morning. And the good tea is through the entire day long. And I still remember, Dr. Tiwari, when you brought me some really fantastic tea from India during one of the conferences. That was a very heartful gift. I appreciated it very much. Thank you.
Most of the Indians, they like tea rather coffee.
I don't drink coffee at all. It's tea 24/7, literally 24/7. And I educated myself. I know that there are different geographical areas in India. There are different altitude.
Yes.
And there are even different time within the tea harvesting season. And you can feel difference with the same type of tea, but harvested from different areas, different altitude. And 1 week is different from another week from the same region. And it's fascinating how much you can appreciate all these varieties of taste and flavor delivered to you by the nature. And, this is really, truly enjoyable. So, I think from what I saw in the beginning during our official ceremony, there were give and take 40, 50 people today. And I hope we will keep audience at least at the same level. And of course, we will appreciate the word of the mouth. If any of you will find our gathering interesting, to pass the word to your friends, to your colleagues, to other students. Who knows, maybe it's something that I will be talking about will raise your interest in the topic. Some of the sessions will be heavily loaded with the handouts, with the slides, and I would like to apologize in advance that we cannot walk thoroughly in the details of all the slides, but I was considering whether I should delete or I should still keep them. And I decided to keep them. Why? For the sake of the audience. So even if we will not have a chance to walk through all the slides, and it's not concerning today's gathering, it's more to the middle of our sessions, at least you will have it in your possession. At least you will know what are the important things and something that may catalyze your interest in the future. And of course, as Dr. Tiwari mentioned, we can communicate either after the sessions or verbally, or you can write the messages, and hopefully later, of course, through Dr. Tiwari, we can communicate as well. And who knows, maybe that will also ignite some fruitful collaborations for Dr. Tiwari and for all of us. gastrointestinal health. So we will introduce ourselves into the topic, and I just mentioned that we will have to talk about microbiota and microbiome. It's unavoidable because we need to understand what these new microorganisms new to the environment are doing and how they communicate and interact with the bacteria sitting already in this environment. We'll talk about the health and disease status and how the resident microbiota is changing under these conditions. So we'll look at the probiotics for gastrointestinal tract and their functions. We'll get some examples and recommendations of American Gastroenterological Association, and that actually is particularly important for the medical application, not for the over-the-counter. When we're dealing with the over-the-counter use, we're pretty much on our own. And then we'll have some conclusions. So we will start with looking at the spatial and temporal aspects of intestinal microbiota. And obviously, we do know that the intestinal microbiota is not homogeneous. And we have different numbers of microorganisms. And you can see it in the chart A, residing in different geographical locations in the equation of the gastrointestinal tract, from around 10 to the 1, 10 to the 3, stomach duodenum, raising to 10 to the 4, 10 to the 7, in jejunum, and flourishing at numbers up to 10 to the 12. That's in the column. And the microorganisms are different not just in numbers, but also in the species and genus that are presented in different ecological niches of the gastrointestinal tract at different sites. So we have the longitudinal heterogeneity, but also we have latitudinal varieties of the microorganisms, and that is something that is depicted in the part E of this slide. And intestinal epithelium, obviously separated from the lumen by the very thick, complex physicochemical functional mucus layer. And the microbiota that is residing in the intestinal lumen is different, very much different from the microbiota that is attached and embedded into the mucus layer. And of course, there is also different microbiota which is residing immediately close to the epithelium. And now we're moving to the timeline, and that is the C part. And I should mention that the temporal aspects will be addressed more in greater detail in a few upcoming slides. And it is also changing through the time. So it is believed that the first microorganisms are acquired through the passage through the birth canal, and of course we do know that the infants, the babies delivered through the C-section have very different resident microbiota as compared to the children delivered through the normal birth canal. And obviously, first microorganisms infants are getting, they are getting them from their mother, right? And then what we see here is that, through the age, through the maturation, through growing, the resident microbiota is becoming established in the environment, stabilizing, delivering its functions, and then closer to the end of the life, we have also some dramatic changes occurring in the eukaryotic host. What is very interesting to mention here would be that the microbiota was studied in the humans in the twins, and both in the monozygotic and heterozygotic twins, and it was shown that it was very much similar. And of course, it's another confirmation of the fact that the primary microbiota was delivered from the mother of the children. Some of the major functions, as they can be assigned to the gut microbiota, obviously it should fight against intruders. It should also modulate the immune system. There are some metabolic roles assigned to the gut microbiota, salvaging of calories, production of short-chain fatty acid, vitamins such as K and folate, participation in drug metabolism, the conjugation of bile acids are also other essential functions. Now, I promise that we will look at the time frame in the closer detail, but this is not the closest I wanted to drag you to. This is where I want to look at other factors, and they are highlighted in the bubble here, affecting the microbiome. And I put a note here: poor skim needs to be explained. Not poor skim, of course. It's already presented in the sense that some may think that these kind of factors, influence a particular time frame, which is not true, of course. Genetic factors that are acquired from the parents who will influence through the entire life. Birth root, on the other hand, of course, should go not to the 3 years old, but to the birth time of birth. Geography will influence through the entire life. We can be born in one place on our planet and then spend childhood in another place, and then our mature life in the third location, and different locations, different climate, different nutrients, different eating habits. All of these, of course, will have a very serious impact on the resident microbiome. Personal hygiene, of course, is an important issue, which comes with the habits and traditions and what people used to do. Stresses, a variety of stresses. They can be stresses associated with the professional activity. There can be stresses associated with the health status and social activities, you name it. Varieties of dysfunctions which come to the stresses, and last but not least, drugs. And the drugs, of course, unfortunately, we're not taken just when we're approaching the mature age of our life, but we can get sick in our adult life, active adult life, and unfortunately also in the young ages as well. So I wanted just to bring your attention to these factors. And now I want to zoom more on the temporal development of microbiota in humans, zooming into aging. And this is where the number one, and I put here the numbers to make it visible for all of us. So the first thing that is happening, the rapid rate of expansion in diversity. Some authors are saying that by 1 year old, the gut microbiota is more or less established, and with high degree of certainty, we can say that like 5 years age, it's definitely established. Sorry for the move, wrong button. And so in the early childhood, the microbiota is moving from the condition of being imbalanced to more balanced, more stabilized, and the bacteroides, microorganisms, butyrate-producing bacteria are establishing their presence in the microbiota. Then by pre-adolescence, which is between the ages of like 7 and 12, I believe, the number of the taxonomical representatives in the gut microbiota is coming to the mature number, and they are of course functionally different. In pre-adolescence as compared with adults, the gut microbiota, for instance, is enriched with anaeroborax, bifidobacteria, fecalibacteria, and lactospiraceae. And this is important that through the time, the predominant microorganisms in the environment are changing. So healthy adult microbiota are usually dominated by the representatives of bacteroides and firmicutes, but also include the representatives of other taxonomical groups such as actinobacteria, proteobacteria, some methanogenic are here, and some other microorganisms. At the high level, the gut microbiota in adults, if you compare with infants, is more stable. And again, we're talking here about the healthy individuals. But some species, and especially strains, will vary significantly among different people. And in fact, we can say with certainty that the collection, the cocktail of microorganisms in each individual to some degree is unique to this person. And despite of the taxonomic variabilities, the functional capacity of the gut microbiota of an adult person is quite consistent across the healthy individuals, with the specific pathways involved in metabolism, in fermentation processes, in oxidation processes, in the biosynthesis of lipopolysaccharides, and many other events. When the age is declining in the elderly people, the gut microorganisms become more unstable, and the diversity is also declining. And that is actually affiliated with all the other changes that the eukaryotic host, the human subject, is undergoing through the aging process. And of course, it is important to mention that it also correlates with the decline in the immune system. We're going to look now at the microbiota of GI tract, and briefly to look at the performance and the status in the health and in the disease. And we're not going to talk in the detail about each of these specific contributions and activities, because this slide alone can take an hour to walk through. And just randomly to mention some things that should be considered as exciting, such as, the role of microorganisms. And here we can see bifida and clostridium, but of course some lactobacilli are involved as well in the production of short-chain fatty acids, influencing the health-promoting roles of nutrition. We can see that the microorganisms, and here again an example would be bifidobacterium, can be influencing the angiogenesis and the behavioral status. So all in all, the different gut microbiota components, they can and they do affect many aspects of the host development and life, while microbiota as a whole often is functional redundancy. And just to mention, some coding in the slide that we see here in a gray, we have the members of the microbiota, some of their components, and I say that not all of them are identified here because we have limited space on the authors who put together this beautiful picture. They had limited space, and some of the components that are produced by these microorganisms are mentioned as well. And in white, there are the effects on the host that are caused by these microorganisms, and in black we have the affected phenotypes. And again, just to read only some examples I give here. And one of the examples I came across as striking my attention, because before reading this paper I didn't think about it. And this is really very interesting, and I put it, with this mark, not to forget to talk about it. Oxalate nectar formigenes, and it has been studied as a probiotic, which, and again, it comes to the question asked today whether lactobacilli are the only probiotic. Certainly not. Bifida is the second, sporoformis is the third. Big groups, but we have some fascinating representatives outside of these groups, and oxalate nectar formigenes is one of them, and can degrade dietary oxalates, reducing urinary oxalate excretion, which in turn promotes successful use in clinical trials in therapeutic and prophylactic options, in calcium oxalate nephrolithiasis and associated renal failures. So, very interesting, unique ability of the microorganism to degrade oxalates turns to be a potent trait that can be utilized for clinical use. This is a very interesting example. I'm pretty sure that we can find similar examples from other microorganisms. Zooming out even more than we did before, we're looking at gut microbiota and what and why can get wrong, and we're coming from the common ground hypotheses, which is depicted in this beautiful picture, and this hypothesis still needs to be proven, although many things look reasonable and logical, and have been proposed to explore the question whether imbalances of gut microbial communities are a consequence or a cause of chronic polygenic diseases. And I know that at this stage it sounds pretty much as the question of the hen and an egg, which one was the first, but aside of the curiosity, it is indeed a very important question. So, what this hypothesis assumes is that first of all, various endogenous, exogenous factors, or the combinations of these factors, can trigger an increase in the gut permeability, causing the so-called leaky mucosa, or the inflammation of the mucosal tissues throughout the selective pressure on the gut microbiome. Then are genetically susceptible to one or more chronic disorders, and we know, a variety of the things, including the gastrointestinal disorders, which can be inherited. The subclinical intestinal abnormalities may favor the expansion of opportunistic microorganisms, opportunistic infections, and then what will happen is the gradual transition to pathobionts, which brings us to the third step, and that is where the microbial gene products from these biotic pathobiont gut communities promote local or even systemic functional and morphological changes that are pathogenic for the microorganism and for the resident microorganisms and for the environment, for the eukaryotic host. And finally, the first step, and that is when the disease-associated microbiota is established, and that is not necessarily just the pathogens, which is almost never happening. It's the combination of the resident microbiota with the pathogens which are dominating the scene, the action, right? And so in the genetically susceptible person, it will be especially devastating, and then, for instance, they can be transferred to another genetically sensitive recipient, and we can see the continuation and the spread of these pathogenic mechanisms causing the infection being passed from one individual to another. So this is a possible scenario. The composition and health effect of predominant human fecal microbiota. Why is that important? Why are we talking about the fecal microbiota? Well, several reasons. One of them we already mentioned, that fecal transplant therapy is on the rise with really good results, with isolated cases when something went wrong because of the samples being misplaced or other things which are not related to the quality of the transplant that was properly prepared for the delivery to the recipient. Okay, this is a very important message. That is number one. Number two, the important thing is that often we judge about the outcome of the therapy, about the outcome of interactions that are happening in the gut, about the ability of the microorganism to establish its presence through colonization and many other things by what we observe in feces. Of course, it is not exactly what is happening in the colon or other geographical locations of the gastrointestinal tract, but to some degree it allows us to see what kind of processes occur here and there, and how the resident microbiota is influenced by the health status of the individual and what probiotic bacteria can do to improve the health and to modulate the health. So normally in the healthy individual, microorganisms and the eukaryotic host have the symbiotic relations, and as I said before, they are trying to reach an agreement, almost as if they're like, okay, well, humans we can think logically, but I'm also talking about microorganisms. Often thinking they have tiny little brain and can think and can talk and can argue, or they can't talk. Warm sensing will be talking about it later in time. So the host is providing with the environment that is rich in nutrients, and on the other hand, the diversified microorganisms, they produce beneficial substances for the host. And used at the non-pathogenic microorganisms is very important. It's crucial for the health maintenance. And even once it is disrupted, the relations between the host and microorganisms can turn bad, even ugly, as we saw in our previous session, with even probiotic bacteria becoming harmful. So this is how the pathology conditions and unfortunately often develop. All right, so I will walk us through the scheme. Bacteria cause pathogenic effects, beneficial effects may lead to other harmful or beneficial effects, and I'm in a kind of a disagreement because we did see examples of lactobacilli LGG strain being off, and of course it should be positioned. You can see number 8, all right? And numbers indicate the numbers of the microorganisms in the logarithmic scale. 10 to the 8 of lactobacilli may be present. Okay, they should have the same location as Escherichia coli. All right, bifidobacteria can cause sepsis. I recall just one example which was not confirmed. So I really don't think so. Bacteroides, yes, can be both. Streptococcus can be both. Enterococcus can be both as well. All right, and bacilli can be both. Bacillus cereus, Bacillus coagulans, Bacillus subtilis, and cereus can be health promoting, but can also cause diarrhea and other diverse health effects, depending on what is happening with the pathogens, the genes, if they're present there or not. Gut microbiota and specific commensals, or perhaps probiotics as potential preventive or therapeutic agents. And we should keep in mind always that the commensal microorganism can become a probiotic bacteria. All right, and it can become probiotic if it is isolated, characterized, multiplied in numbers, and delivered de novo as a supplement to the eukaryotic host who may need these microorganisms to modulate one another health condition. All right, and we can see here, well, stool transplantation. Of course, stool transplantation transplants are not considered as probiotics. We'll see it when we'll be talking later on about other things related to regulations and definitions. But of course, you can see a variety of applications where the microorganisms can be used, and clinical studies are indicated in blue, and clinical mouse studies are in red. And basically, this is what we see with a variety of microorganisms and their possible applications. And actually, a variety of studies, the findings suggest that microbial-based therapeutics or preventives, depending on the role they play in the strategies, they may have advantage over the use traditional approaches such as different medications, or they can even be used complementary together with medications. And important thing to mention that probiotics may cause, and often usually cause, less side effects, less non-desired effects on the human subjects when used in a variety of treatments, and close to none negative effects are observed in the animals. Gut microbiota function are associated with a variety of diseases. And as I mentioned before, we should consider ourselves as a combination of ecological niches, but not which are populated by microorganisms. But we are not just the niches, we are also a collection of the functional mechanisms which are functionally connected to each other. And the interesting thing that these mechanisms are also influenced by the resident microbiota, but most importantly, they're influenced by the gut microbiota, which act locally and systemically, as we mentioned. So, when we're looking at the pathogen-host microbiota interactions and the outcome of the infection, several things that we should keep in mind. The enteric pathogens, they utilize their weapons, their arsenal of the virulence factors to fight against the host response, and that destabilizes the local resident microbiota and affects in many ways the protective and immunomodulatory function. So what we have is things that are attacked: the resident microbiota and the host itself. The resident microbiota is attacked on numerous levels. It's pushed away from its ecological niche. And the first thing that needs to be done with the host is to destroy or to at least weaken its immune system. In addition to that, the pathogens most likely also interact directly with the microbiota through many factors. The arrows leading to question marks indicate that the factors are still either unknown or need to be thoroughly studied. And as a result, these interactions, the pathogen can multiply in numbers, can establish its presence in the environment, and can have the further negative impact on the host and the microbiota, which may also result and does result in the post-infection microbiota instability. And we're talking here about the condition that may persist for the significant duration of the time after the treatment was applied, and after, for instance, the pathogen is eradicated, after the eukaryotic host is returning to its normal healthy status. So we should not neglect the need for the restoration of the normal resident microbiota. Just as an example, the role of microbiota in inflammatory bowel disease, pathogen disease, and this is where we know for a fact that probiotics may and they do help a lot. And the interplay between the host resident microorganisms and host genetics is very important, as we mentioned before, and some of the parameters here are not presented in this figure, and the authors didn't do it not to overcrowd the image. And that may result in the progressive inflammatory damage to the host intestinal condition, to the host mucosa, manifested through the observed characteristic histopathological findings and symptomatically. So what we have here is in the purple boxes examples of genetic deficit, green boxes examples of all kind of anomalies, boxes examples of histopathological findings, yellow examples of symptoms, and red arrows possible clinical course of IBD. And we can see here that, at each of these stages, the role of the resident microbiota, and should the probiotics be used, is difficult to overestimate. You can see the change in the composition. I'm bringing your attention to the point 2, where we have the change in the profile of the microorganisms. And while the drug treatment is usually applied, it is believed more and more that microorganisms can, as probiotics, can significantly contribute to the establishing of the healthy resident microbiota. So how probiotics can act in the gastrointestinal tract and what they can do. And some of the functions you can see on the left is simplified boxes. On the right is, I wouldn't say it's over-engineered, but comprehensive picture. And of course, some desired things, probiotics can help with inflammation, and of course, all diseases, lactose intolerance. I take it off the picture because nowadays there are a big variety of dairy products that are lactose-free. And being myself, to some degree, lactose intolerant, of course, I like dairy products. I like cottage cheese made in a traditional farm manner. And I know that I will suffer, but I will take a spoon or two of you once in a while. But there are lactose-free products which have good taste and, very much indistinguishable from other products, which are still not labeled as lactose-free. So I will take these parameters chart. Again, you can see here some of the features that are desired, some of the features that may not necessarily be desired, such as adherence and colonization if we're dealing with a transit or the microorganisms that are not colonizing at all. Production of bacteriaceae and weak organic acids or hydrogen peroxide are really potent weapons at hand of the resident microbiota and probiotics, which help in a variety of ecological niches, starting from the mouth and going down to the colon and other organs and tissues, vaginal environment, to name just one, and many other. But one of the things that you can walk through all these things and all these, I call them geographical locations of the human body at different phases, and the parts. So we have the compartments, and we have the stressors, so harsh conditions as they call it in this figure. And the stressors are varies pH, salts, acids, enzymes, and many others. And all of these, the microorganisms that are delivered to health, they have space through their massage and action through the gastrointestinal tract. Now, one of the regions I highlighted here for very special reasons. All the rest, we take as given. But, unfortunately, not that often we see in the papers oral cavity being mentioned as the beginning of the gastrointestinal tract. And this is where the food digestion actually starts. This is where the food uptake actually starts. This is where the probiotics may start working as well, and they do start working as well. Needless to say, and we will address this issue briefly later on in one of our upcoming sessions, that the oral health is intimately linked to the cardiovascular health. So these things cannot be separated, and we need to learn more about the possible impact of the oral health on other organs and tissues, not only cardiovascular one, which came as an obvious thing, of course, some years ago. So, in the mouth, when probiotics are orally ingested, they are initially exposed to saliva. Okay? And saliva is the secretion by the salivary glands, and it's a complex fluid, a with a pH which can vary from around 6.2 to neutral and slightly above to 7.4, I think. And it consists of mainly water, about 94% of water, mucus, which is proteoglycans and glycoproteins, some electrolytes, enzymes, including amylase, lactic peroxidase, lipase, lactoferrin, and lysozyme can be present there as well. And I cannot emphasize these enough for the very obvious reason. They are also antimicrobial, and they play a very important role in antimicrobial properties of saliva. So they may act against pathogens, but they may also stress the health-promoting microorganisms. This is something that has to be kept in mind. So if you design a new probiotic that has to work in the oral cavity, of course it should be resistant to these kind of substances. Also, saliva may contain, does contain some amount of hydrogen peroxide, also not very high, and immunologic components, which may include IgA, IgG, IgM, and there are some other defense substances, defense mechanisms encountered in saliva. Some of them to be mentioned here, like antimicrobial proteins, antimicrobial peptides, such as cathelicidin, defensin, and last but not least, certainly histatins. And histatins are called that way because they're histidine-rich antimicrobials. And some authors actually, these histatins are very active against a broad range of microorganisms, including yeast. And some authors indicate that dry mouth syndrome, for instance, which is observed in compromised IgV individuals, these individuals with the dry mouth syndrome, they have oral candidiasis developed, and some of the authors indicate that perhaps this is because insufficient amount of saliva, insufficient production of histatins, which may control pathogens in this environment. Main mechanisms of action for probiotics in the gastrointestinal system, again, we're looking at antimicrobial action, we're looking at immunomodulatory action, we're looking at the interaction with the host, and in general, 3 potential levels of interaction can be identified. So, number 1, and that is within the gut lumen by direct interaction with the local ecological system of the gut microbiota. This is the most important thing. And probiotics can also have a direct metabolic impact in the gut. They can provide with certain enzymatic activities. For instance, you mentioned Bacillus natto and the ability to produce nattokinase, and nattokinase may help, for instance, to regulate the blood pressure, among other functions. The second level of interaction is with the gut mucus layer and epithelia, including the barrier functions, involvement in the process of digestion, acting at the level of the local mucosal immunity, and interacting with the enteric nervous system. Speaking of the integrity, it has been shown in a couple of papers, unfortunately not that many, but I hope these area will be developing. It has been shown that, for instance, bacilli, particularly Bacillus subtilis, can have beneficial effect on the integrity of the epithelial layer of the cells. And we should keep in mind it's just a single layer of the cells which provides our gut with such an important protection. Finally, last but not least, when talking about the interaction through signaling to the host, beyond the gut, to the liver, to the systemic action of the systemic immune system and other organs, tissues, all the way to the brain and back. So these are if to outline in a short, in the main mechanisms of interaction. Direct effects of the intestinal epithelial cells is something that comes first, and that comes with the increasing of the secretion of mucin, glycoproteins by the goblet cells, and these goblet cells assemble into a thick mucus layer. Then we have the secretion of antimicrobial proteins, or you mentioned defensins. And defensins, proteins, they are produced constitutively. And they, some authors actually consider them as the first line of defense that starts working even before the actual immune system is responding to the intrusion of other changes in the eukaryotic host. Right? And they may help, they do help eliminating the intruders and the mucus layer. Then we have the enhancing the stability of intercellular junction complexes, which are known as tight junction, which decreases the intracellular permeability of EC to pathogens and to antigens. The inhibition of inherent pathogens occurs through the introduction of a variety of antimicrobial substances. Some of them already mentioned today: weak organic acids, hydrogen peroxide, and last but certainly not least, bacteriacids. And we will have, as I already mentioned, a standalone lecture, a session dedicated to bacteriacids. And of course, the competition is observed with the commensals and pathogens, the competition on many levels, the competition for the attachment side, for the adhesion, for the nutrients. So all these competitions are actually utilized in what is known as a strain replacement approach, where the probiotic microorganisms can be used to push the intruder away from the ecological niche, just because the probiotic bacteria are stronger as competitors on many levels. And probiotics can also, and that is not shown here, can also alter the natural gut microbiota composition or the expression of the genes, and this way they can also work on the improvement of the barrier integrity with assistance of the commensal microbiota. Now, speaking of the role in the change of composition of the gut microbiota, it can be good and bad, and we will have one of the examples mentioned in the upcoming session, because it has been shown in some cases, in some studies, in poultry, I believe, that the probiotics may also increase diversity of the resident microbiota without causing any phenotypical negative effects in the studied animals. So whether it was harmless effect or not, still to be understood. But, they can work both ways. And of course, since we're dealing with the protection, the causal immunomodulation by probiotics is a very important issue. And I told you that before we started that understanding endocrine system and immune system comes hand by hand with understanding of how probiotic bacteria and commensal microorganisms work in the given environment. And obviously what we observe here, A through D, is the downregulation of the pro-inflammatory cytokines such as interleukin 8, and the upregulation of the modulatory factors. So all these processes take place through the signaling by probiotic bacteria, such as, for instance, probiotic may signal with the interacting with the dendritic cells to stimulate anti-inflammatory cytokines, as I mentioned, such as interleukin 10, and that can be a result of the answer to the intestinal infection, right? They can also have the levels of IgA secreting plasma cells in blood and propria, and through that promote the transcytosis of the secretory IgAs through the epithelial cell layer and the secretion into the mucus layer. And these can assist with the prevention and of the penetration by the non-desired microorganisms to the human tissues. We're changing the gears and going to the next topic of this session, that is the probiotics for guys, with examples of studies and some recommendations, and if you wish, restrictions by the American Gastroenterological Association. And while of course the temptation is high to spread the use of probiotics all over the environments and niches in humans and animals, we have to be careful. And when we look at the strategy, the summary of the strategy for searching evidence and selection, that comes from 2017, but it didn't change really much. I'll just drag us all through one of the examples, but they're very similar. All right, and we start with the first primary selection of more than 1,000 cases, and then we narrow them through the selection of particular conditions, such as, I don't know, randomized control trial, clinical trials on the 89. All article titles of that will be 200. All right, and then when we go from that, we narrow to the specific conditions. And for instance, for IBS, we have only 61. So it means that although, again, the take-home message, and I always try, whenever we have a comprehensive table, flow chart, or picture, or figure, to bring attention to the take-home message. And the take-home message is that while there are many studies being conducted, there is huge diversity in the ways how these studies were conducted, the specific details, the outcomes, and what was really studied. And so quite often, if we're not comparing apples and oranges, we're comparing green apples versus red apples, sweet apples, non-diet sweet apples. And that's something that makes it challenging to collect sufficient data for making really sound and solid recommendations. So when we look at these examples, systematic reviews and meta-analyses, and what kind of outcomes we have. And again, in my own pretentiously handwriting, of course, it's the computer-generated style, major take-home messages. They're safe, and they may work. And when we look at the diseases, diarrhea, either the acute infectious diarrhea, or traveler's diarrhea, or C. difficile cause diarrhea. Yes, they work in many cases, and we can see I highlighted the ones where some effects were shown in the reduction of symptoms, reduction of the duration of the time of the unhealthy condition. But there are also some conditions, no sufficient tests to support the use of probiotics. And I'm extremely sad and unhappy because what I see here is the strains, the microorganisms that I personally like very much. We have Nisla 1917, the one that I mentioned before. We have Lactobacillus GG strain, and they didn't do any good job there either. So it depends. And what it depends on is still to be studied, identified, and understood. And I think the biggest problem here, and we'll be probably talking about it again when we'll be dealing with the probiotics for animals. Also humans are taxonomically also animals, but we're different animals. We are the creatures who are more diverse in every possible aspect. And being that diverse is a luxury, a gift, and a burden, because the microorganisms can work one way in a certain group and absolutely differently in a different group, or even different from individual to individual. A generalized scheme for the mechanisms by which probiotics work is a kind of a tri-dimensional communication, which is involved between the target host, the commensal microbiota, and the probiotic. And most likely, a variety of diverse mechanisms are involved in what we see as the health promoting action of the probiotic bacteria on the host cells. Hello? Somebody is trying to say something. Okay. In some cases, such as production of antimicrobial substances, cross-feeding other microorganisms, these kind of mechanisms are driven directly by interactions with the resident microbiome. And in other cases, when we have direct interaction with the immune cells through the signaling, of course, the effects may be directed through the interactions with the host organism, the host cells. And overall, we can say that the clinical benefits delivered by probiotic usually can result from the combined action of several mechanisms. And while multiple mechanisms probably coexist and co-express in a single probiotic, the importance of specific given mechanisms will depend on a variety of factors. And some of them are actually highlighted here, some, but certainly not all of them. What are the take-home messages here? The take-home messages are simple and straightforward. I hope that the human gut microbiota is integral to health and associated with a variety of diseases at the same time. And the effect of probiotics, either as a prophylactic measure or as a therapeutic agent, occurs through the modification of the microbiota and its function, and it also occurs through the direct action on host through the interaction with the variety of tissues and organs and systems. Most importantly, through the immunomodulation, through the signaling, through providing the barrier function and the formation of enzymes, and the last but not least, through the interaction with the endocrine system with the central nervous system and the brain. Coming to the suggestions and advice by the American Gastroenterologists Association, it doesn't recommend the use of probiotic for most digestive conditions. What it means is that, well, if you're buying something over the counter, you're on your own. But, for the treatment by the medical professional, most likely you will not get probiotic as the major drug for treatment. Nevertheless, the American Gastroenterologists Association acknowledged some of the benefits, such as, for instance, the particular specific probiotics can prevent mortality and necrotizing enterocolitis in the preterm children of the low weight, and especially in the hospital treatment. And we have some fascinating examples of probiotics saving children. One of the examples would be in the Yakult case, which was documented, and I have a video kindly provided to me by Yakult company, and that was absolutely fantastic. And the level of miracle when the child was delivered with many anatomical challenges, passing through several surgeries, treated with antibiotics as a preventive measure to avoid any infections, either post-surgical or otherwise, and having no resident gut microbiota. Everything was replaced with Candida, just Candida, and probiotic formulation was delivered to the specifically designed by Yakult Institute, was delivered to this child, and the normal microbiota was restored in the kid. This is really, although I understand to some degree the science behind these processes, I still take it as a miracle. Probiotics. I'm sorry, whoever is talking, please mute yourself, all right? Certain probiotics, according to the American Gastroenterologists Association, should be considered for the prevention of the C. difficile infections in both adults and children. And at the same time, probiotics do not appear to be beneficial for children, at least in North America, who have acute gastroenteritis. And there is an insufficient evidence, according to AGA, to recommend probiotic treatments for the infection with C. difficile, for Crohn's disease treatment, for IBS, ulcerative colitis, and basically they don't recommend the use of probiotics. And I doubt in this case insurance companies will cover the use of probiotics. But interesting thing, Crohn's disease, we will not talk much about the Crohn's disease in the future. I think not at all, but I should probably mention that somehow it carries some resemblances with jaundice disease in cattle. And in jaundice disease, mycobacterium tuberculosis most definitely. And in Crohn's disease, there were some reports indicating possible presence of mycobacteria, not tuberculosis, sorry, mycobacteria in the biopsy samples. And if that is the case, if mycobacteria are associated with Crohn's disease in one way or another, there is a good chance that mycobacteria can be addressed with probiotics. We're coming now to the conclusion of our gathering. We're almost in time, which is good. And where we're dealing today with the probiotics in gastrointestinal health and resident microbiota. And if we will take a look again, so from the GI symposium, the third symposium where this picture derived from, we have a variety of conditions that can be addressed H. pylori eradication, I'm not sure, but I hope it will work because these microorganisms that cause disease, colitis, diarrhea, and many other conditions in gastrointestinal tract, that's disease. I have confidence in saying that they probably will be controlled with the probiotic bacteria. And so looking at the walk through the our lifespan and what microorganisms do, and where the health-promoting bacteria can be involved. I'm giving you the last picture. I'm very proud. It's not from my publication, but I was communicating with the author, and he asked me, what should we do? What kind of a special twist we should give to this paper to make it eye-catching? I said, why not to give a picture which shows the walk through the life and communicating, with the environment and telling the story, what happens with the microorganisms from the condition what's imbalanced and just a few first bacteria coming, thanks mom for them, and moving on, communicating with other humans, getting them from the environment, and then passing through the maturation and to the elderly stage, and complaining if I would only have more for kids. And I think it's really important to also have it as another take-home message. When we design probiotic supplements, they should address not only the ecological niche, but they should also address the age stage of the human beings if we're dealing with probiotics for humans. And with that, I would like to finish out today's gathering, and I thank you all for your attention and for being with us at this late hour. Thank you.
Thank you, Dr. Chikindas. You have very effectively communicated with the kind of probiotics can be isolated and what are the features, how we can identify. And it is very difficult to identify their behavior. Sometimes it differs mostly from one individual to another, from one niche to another, from one ecosystem to another ecosystem. So it's a kind of fishing from the ocean that under what condition is they are going to benefit. Some of the students, they have written their questions, so I'll quickly read you to you.
No, I don't, Dr. Tawari, I don't see the questions in front of me, so I will.
Okay.
Yes, I have them. So, and I have the timeline. So I would allow myself to answer in a chronological order. And again, don't take it as the last word carved in stone. Of course, I'm a human being, so I will be talking from my experience and my understanding of the subject. And yes, that is a kind of a legal disclosure, if you wish. Okay, can probiotics cause addiction? And I may see where this is coming from, because with addiction, even the food can cause addiction. Some are saying that cheese can cause addiction, and just to put it in a humorous manner, I'm a person addicted to cheese, seriously. And if we dig deeper, we find certain substances that are identified as possibly causing addiction. To the best of my recollection, I don't remember any papers published saying anything about the addiction as a result of the use of probiotic. Another question is the formulation. And we know that probiotics are grown somewhere. Some of them are grown in the milk, some of them are grown in some plant substrates and other substrates. We must remember that the meat origin substrates should be avoided when we're dealing with the industrial production, and that comes with safety regulations. How the constituents of the delivery system and whether they may cause addiction, that I know. All right. Food interaction with probiotics, any harmful cause? Well, harmful in a the sense of harm to the food product, and in the case of possible harm to probiotic, I'm guessing. We don't see these kind of examples. However, when the probiotic is designed for the formulation and delivery through the food product, of course, we need, and there are two ways, either by mixing, expecting that the probiotic will not propagate, will not multiply in the products, or probiotic can be used as a part of the consortium of microorganisms involved in the fermentation process. In the first case, obviously, what we're trying to do is to prevent the microorganism from multiplying in the environment, and that can be done through a variety of formulations or through the control conditions such as temperature or the storage of the food product. And the second option, when the microorganism, probiotic microorganism, is a part of fermenting consortium, obviously, all the studies have to be done to make sure that the microorganism survives in the matrix. It can also live in the peaceful coexistence with other microorganisms, and it doesn't change any rheological taste, flavor, and other qualities of the food product. Is there any probiotic for cancer? Revert back. I'm not sure what you mean by revert back, but I don't recall any probiotics studied in particular for oral cancer. For a variety of oral and dental conditions and disease, yes, by all means. And I would refer you to the studies, especially conducted and still running by University of Otago in New Zealand, and Dr. John Tagg, who is one of the gurus of oral and dental microbiology, especially with the emphasis on bacteriocins and oral probiotics. How about the safety of other strains apart from lactobacilli? The safety, well, we will be talking about it in the future, but since the question was raised, my personal belief, it's case-by-case study. Every strain must be evaluated for its safety. Although there is a lobby now by very well-recognized scientists who think that some species perhaps should be recognized as probiotic species. I'm very much against it. I think better to say it. Sorry, but we have other examples as well. So it's not black and white. Right now, it's a gray area. All right, on a different scale of examples. For instance, in the US, there is a zero tolerance for Listeria monocytogenes in ready-to-eat products, period. In Europe, there is no such regulation. And one of the arguments can be, well, we know that women, even pregnant women, can be carrying Listeria monocytogenes asymptomatically with no diverse effect observed on the fetus or the delivered child. I don't know. When to take or not to take probiotic? Whenever it pleases you. But it's case by case. Probiotics may or may not help you. You never know. I told you the example with my own, Activia, and I have this cardboard on my desk which says about, fastening the passage through the intestine, and that's exactly what it did to me, causing diarrhea. 3 times I tried it. We don't want it to happen. So it's individual, number one. Number 2, go for the strains that have very good documented history. Go for the formulations that have these strains on the label, not just the species. Study the strains, look in the literature, clinical trials. And as for the time, it's commonly suggested to take probiotic with the food, and preferably not the hot food. I didn't suppress patients getting blood infections with bacteria from the probiotic they are taking. I wouldn't say it in such a generalized manner. It may happen. It may happen. And we have some indications that it was reported in several cases, but I wouldn't say that this is common. Are probiotics helpful in eliminating the side effects of chemotherapy in cancer? I recall just a few reports saying that, but of course, you know that if the negative results occur, often negative results are not being reported. How effective it is if we introduce soil isolate LEP strain in mice? Well, it's nice to see somebody caring not only about humans, but also about mice. I personally love the little creatures. Look, the modern, commonly accepted definition of probiotics does not emphasize the origin. But of course, to some degree, you may be right with your question. In which sense? It is more hoped and expected the microorganism that is isolated from your own environment to act better in your own environment. But it is not always the case. And we will see some examples in our animal session that certain probiotics work well, for instance, in dogs, but not in cats. And certain human probiotics may work also in animals as well. How the probiotics can use on solid food. On solid food, I'm not quite sure I understand the question. Well, they can be mixed through the formulation, added to the matrix, or you can sprinkle them on the surface. All right. I mentioned, just recently passing the probiotic for guinea pigs to one of my friends. And this is just a powder which is sprinkled on the surface of the granulated food for the little creatures, and they eat it. In the case of immunocompromised people, it is being said probiotics act adversely on health. What is the mechanism by which it happens? What is the mechanism by which it happens? What is the mechanism by which the microorganism can move systemically? And I appreciate this kind of question, because every time I'm talking about it, I'm asking myself the same question, and I'm trying to dig the literature in search for answer to the same question. How is the gene expression profile changing in the probiotic that is administered to the immunocompromised host, where it starts behaving as an infectious organism? I couldn't find the answer so far. And maybe somebody is working on it. Maybe something was reported. If you will find something, please share with me. But I think this is very important, not just from the point of view of fundamental science, but it is very important to build the safe application for the problem. Imagine if you know these kind of genes that may turn probiotic bacteria into the wolf, into the bad creature. Imagine if you can engineer probiotics for the medicinal application. Engineered probiotics are allowed for medicinal application. And you can, for instance, eliminate these genes. You can delete them or truncate them, or turn off their regulatory elements, and thus to make them acceptable for applications in immunocompromised individuals with a high level of safety. I think this is very important. No, not only, I want to ask a probiotic use in food safety, mainly solid food like cheese. Not necessarily. I don't recall, we don't have a session dedicated to the applications of probiotics in food, but oh my God, cheese, variety of dairy products, fermented dairy products, non-dairy juices, chocolate bars, cereals. So the only limitation is how to deliver these probiotic alive through the manufacturing process and storage. Oh, tea. We were talking about tea today, and I don't have it right now with me, but I have it somewhere on the shelf in my office. Tea. The tea with probiotic. You can ask, well, how is that possible? Of course, we're not talking about the iced tea, the typical American tea. We're talking about the hot tea. Guess what? Spore-forming probiotics can be delivered with the tea. And this product is already on the market. So there are plenty of varieties for application in many food products. And the only limitations is the delivery system and the approaches that will make the microorganism alive through the manufacturing and storage. Thank you.
Thank you very much, and you have explained each questions very nicely. And hope students will be satisfied. And even if you have any question further, you can email me, and we can discuss in next session whenever Dr. Chikandas will join tomorrow. So not an issue. He is with us for a week, next 5 days. And any idea, any views, anything, if you have in your mind, you can post me, and we can discuss this in tutorial session also, and also in the lecture of Dr. Chikandas. Dr. Chikandas, we have lecture tomorrow at 5:30 at our time. Maybe yours is 7 in the morning.
7 in the morning.
Yes, so tomorrow we'll manage 1 hour, same like that.
Right.
Yes, so we are also having a tutorial. Our time is in the morning from 11 to 1 p.m. There I'm taking them as a virtual tour to Yakult industry. We have an industry nearby. Yakult has a manufacturing unit with us. So they are not going to physically, but virtually they will demonstrate how they grow probiotic. And see, one of the laboratory we have collaborated with the artificial gut, so they are going to demonstrate them how we can have this probiotic from different part of the gut. And in my lab, I will demonstrate like how probiotics can be grown, what are the property of these probiotics, and purification of bacteria at the end of this. So one big tutorial also, these participants will be exposed.
This is fantastic, and as you know, I really am fond, if you can say so, of Yakult. And when I was building a part of the course for my students, I had a really very valuable support from Yakult with the video that I am showing for the undergraduate, not graduate level, but for the undergraduate students with his clinical case I mentioned briefly today. And I really believe in their work with one marketed product and endless varieties of the targeted formulations that Yakult Institute is producing and helping many people in need of restoration of their microbiota, resident microbiota. And as I said, we're planning for the Yakult sponsored meeting in Brazil, probably in 25, I guess. So this is the upcoming in the future event, which I hope I will be able to contribute to the best of my ability. Thank you, everyone, and I will see you tomorrow. And have a good evening. Dr. Twari, we'll see each other again. And check your email and send you the message. Thank you. And bye.
Have a nice day.
Thank you. Bye-bye.
Bye. So from the participants, if you want any query, we will meet tomorrow. We are having 2 sessions. Morning session is at 11. We'll have a tutorial part where you can expose for most of the practical laboratory demonstration, industrial visit, and also also some of the high-end instrumentation demonstration will perform. So thank you for your registration, and thank you very much that you joined our this GIAN course. Hopefully, I think if it is not directly linked with your research, you may find some fruitful discussion, interaction with the exposure of Dr. Jigendas, who is very pioneer discoverer in several areas. You can Google his name and find several publications and patents is his name. So that's the way one can learn, and hopefully I think it will be beneficial for your research work and also in future career, how to shape it in this direction. So we have tried our best to not only give a theoretical lecture in the morning, but also in the tutorial section you will have a better chance to interact with us at the practical aspect. What are the instruments you want to use, and what are the instruments that are actually required for the probiotic growth and probiotic application. We are taking you to the industry. We have talked to the Niti Aayog Atal Incubation Center. They are providing a very good platform where one can launch their idea how to develop a probiotic into a commercial product. So in one of the tutorial section, you will also be able to directly interact with the CEO of Atal Incubation Center. They are having a fantastic support to the students who are working in this area. So with all this, good night to all of you, and see you tomorrow.